So yeah, today I'll give you a brief overview and update regarding the work I've been conducting. This is actually work that I've done partially when I was in Nestlé Research in Switzerland, which then we finished over when I moved to NUS in Singapore to join Brian Kennedy's longevity program over there. And really the focus today is going to be more about this new metabolite called Trigonelline as an NAD modulator for muscle aging. And actually I will give you a sneak peek about some new work that we are submitting regarding kidney disease as well. So just a small disclosure as well, recently I also joined as a consultant the Live Beyond, that is a company co-founded by Brian Kennedy and Yong from NUS, and where Dr. Chong here is sort of leading a little bit the operations. It's more about exchanging on applications of the work that we are doing in NUS in Singapore, and there's other main scientists on the board as well. So if you have specific questions about the work that we do and potential applications, feel free to discuss with Dr. Chong, with me, or with Brian through emails and so on. Okay, moving to the science. So we all know and we are all familiar, I think in this conference has been covered long enough about all the different cellular processes that become dysfunctional in the process of aging. This can be at the cellular level, at the physiological level. And we know that some of this, what's interesting about that is these are cellular hallmarks of aging, or people like to define them as such. But when you are aging, you have aging plays a major risk in aging-associated diseases. And in my case, what I'm very interested in, more going to give you a bit of an introduction today, is about kidney disease, for which actually there is a more increasing global incidence, which is already estimated at 10%, but in the middle age to over 40%, to older age groups, it's over 40% incidence. And if you consider that a lot of people in their middle age and so on also have diabetes, and half of the population that has diabetes has kidney disease, this is becoming a major age-associated condition that needs solution as well. And of course, if you think of muscle decline, there's a global incidence for cohorts age 60 and older estimated to go up to 30%. And if you look at Singapore, for instance, where I'm currently working, this incidence is even higher because, you know, the scores for sarcopenia, the way that we score for sarcopenia is becoming more and more, I'd say, inclusive in a way, and actually the incidence of sarcopenia is increasing much more than what we expect as well. So to move on the first part of the talk regarding sarcopenia. So as I mentioned, sarcopenia is not just a loss of muscle mass, but actually it's described by the loss of grip strength, low muscle mass, and also loss of gait speed. So you need at least 3 clinical indicators to define sarcopenia. And the range of the indicators through which, you know, these are classified then, or they classify you as sarcopenic individual are a bit different between the European consortium and the Asian consortium, but they are clearly indicated. So they are quite close in the range. Now, from what I'm interested in, and with the work I was doing in Nestlé Research initially, what we're interested in is to understand how sarcopenia from a molecular perspective works. So what are the cellular hallmarks of sarcopenia? And if you look at it, actually between muscle aging or the aging process itself and sarcopenia, you will find out that a lot of the hallmarks are indeed shared between aging and muscle disease, as I just showed in the previous cartoon. So for instance, dysfunction of mitochondria is a primary hallmark of sarcopenia as well, increased inflammation, dysfunction of stem cells of the muscles that are satellite stem cells. This is also very prominent, and these are important for muscle regeneration. So with aging, this function tends to decline. Imbalance in protein turnover and increased fibrotic and fat infiltration in the muscle fibers. These are all hallmarks specifically to the muscle, but they're shared with the aging process as well. Connecting a little bit all the different cellular hallmarks of aging, at least from a preclinical perspective, a key coenzyme that has been already described by Dr. Yong and other speakers here is NAD+. So again, preclinical work has shown that basically if with aging in certain tissues or with age-associated diseases in particular, NAD levels may tend to decline. It doesn't decline with the same speed in all the tissues or with the same intensity in all the tissues. So in some tissues seem to suffer much more from this problem. But from a molecular perspective, it's a very clear critical coenzyme, as it's been involved pretty much in more than 500 enzymatic reactions. So it does take place to any pretty much enzymatic reaction of relevance in the cells. We also have to keep in mind that since it's such a critical coenzyme, its metabolism or homeostasis is supported not just by making more NAD, but also how to prevent its degradation or how to support its levels through different interventions. So I'm not going to go through all the interventions because I will focus more on the NAD precursors part, but essentially exercise, calorie restriction, fasting. So lifestyle adjustments have been shown to support healthy levels of NAD. And by, for instance, blocking processes or intervening on aging itself, but also at the enzymatic level, blocking enzymes that consume NAD levels has been proven beneficial to support healthy NAD levels. And of course, if we would have a way to limit or block these age-associated diseases that you can see here, this would be another way to prevent this excessive NAD consumption. So essentially, NAD homeostasis is really a balance between synthesis and consumption. And in particular, what has emerged also from the talks here, or overall also in the last 10, 20 years, is really a large focus on how we actually synthesize NAD. And in particular, we focus and give a brief overview about NAD precursors. So this is a very simple scheme. I think people have mentioned already several of these pathways. There are many, many ways that your cells can synthesize NAD. So it's not just, for instance, the salvage pathway that you have seen here, for which all of you are familiar with the nicotinamide riboside and NNM, as you have seen again from the previous talks. These have become very prominent also on the commercial level because essentially the reactions to get to the NAD are basically 2 steps for NR and 1 step for NNM. But you also have other key molecules that have been historically linked to NAD production. For instance, Dr. Yong just talked about the de novo pathway that starts from tryptophan and it leads all the way to NAD production. And it's in fact important in some specific organs, but it does have a lot of reactions, about 16 to 18 reactions to lead to the NAD production. And then you have, for instance, the nicotinic acid or niacin pathway, which has been historically linked to NAD production. It has been used clinically, and I'll give you an example from some Finnish study in humans with some good success. However, has been, for instance, associated also to detrimental effects such as flushing and gastritis and so forth. And then you have actually a search, so there's a continuous quest for new NAD precursors, and I'll give you an example why, due to the limitations of some of these precursors, where more recent research has looked into the so-called reduced NAD precursors. So you have nicotinamide riboside, or NRH, and NNMH, which do show some advantages in terms of stability, and they have some preclinical benefits, but they are still very early stage in terms of development. So as I mentioned, we know that the cells are producing all these precursors and these metabolites, but from a commercial or application standpoint, people are focusing specifically on nicotinamide riboside or NNM. But I'm just going to give you an example of the pros and cons of some of these metabolites. For instance, nicotinamide, which is also part of the salvage pathway, and by the way, it's generated by NAD degradation, and also gets converted into NNM before entering NAD. Commercially available, it has multiple commercial sources, low cost, but it has some limitations. For instance, the benefits may be NAD independent when you increase the dose, and some negative impact on long-term clinical testing for immune system and the pancreas, for instance. Just to give you a couple of examples. Nicotinamide riboside and NNM, they actually kind of share some pros and cons that are comparable. So for instance, nicotinamide riboside, it's 10 times more expensive than NAM, and I think same goes for NNM to some extent. And the limitations is that for both NR and NNM, but more for NR in this case, is that it's unstable in liquid solutions, and it does get degraded to NAM in the gut and circulation. So once you ingest these molecules, your gut microbiome will metabolize these compounds, your liver will metabolize these compounds, and you actually end up in your body with a mixture of NAD precursors. You don't end up only with the one that you are taking orally, for instance. And this has also steered the interest in things like sublingual or IV infusions, which are still very early stage and we're not going to discuss today. And if you think about these alternative precursors, such as NRH or NMH, yes, they are much more stable than NR and NNM, but there are still issues related. They are very recent molecules, so the scaling up for synthesis is still not optimized. I mean, there are a few companies starting to sell them, but also the safety studies are still needed. There's no official clinical trial that has been published showing concrete benefits of these molecules, and the preclinical studies are also still limited. And just to give you an example from our own metabolomic studies, if you, for instance, put NR and NNM in a human serum at 37 degrees, just without cells or anything, just in a human serum at 37 degrees, and you do a time course, you will see that over time, by 2 hours, they're going down, and by 24 hours, they're completely gone. And what you're getting essentially is a release of NAM, because NR and NNM, they contain a NAM unit. So in a way, just to give you the idea that once they're in your body, you don't just get NR and NM, you're actually getting a lot of NAM, which also your gut microbiome will convert, for instance, in a nicotinic acid. So again, just to show pros and cons of these molecules. And when we look at the clinical translation, as I mentioned, some of these are already been applied, and there is some benefits. I mean, there is some emerging clinical evidence that some of these compounds are starting to bring some benefits, but they are still in the context of not so much healthy longevity or healthy aging, but in the context of age-associated diseases or mitochondrial dysfunction. For instance, here on the left, I'm giving you an example of a positive application of nicotinic acid, which actually was used in the context of mitochondrial myopathy. So again, these are not healthy elderly. These are individuals that are born with a genetic disease that causes mitochondrial dysfunction. And why it's interesting this, because these people really show impaired muscle function at very early stages of life, and it's really impairing for their life quality. So here, this group in Finland with aiapirinin, they actually use niacin as a press-handler substrate, and what they showed is that you can treat patients for quite even long-term trials, and it's well tolerated, and most importantly, you can actually improve significantly phenotypes of muscle health, which would be quite limited in these patients otherwise. Yes, then the thing is the trial worked pretty well in this context, and I think it's been scaled up for other applications as well. However, there is still limitations related to the fact that the molecule produces this painful flushing and other side effects. So unless you are very compromised like these individuals, you may not want to consider that as an healthy individual. And of course, as I mentioned here, this is not an healthy aging case of trial. This is essentially a pathological condition. And if we think of NNM, right, that it's already available on the market, I mean, I guess many people are using it already daily. Yes, there is some first studies here from, this is work also from Shinichiro Imai and so on, who is a big representative of NNM, showing that in some cases where there is decreased, let's say, insulin sensitivity in pre-diabetic women, for instance, you can actually improve insulin sensitivity. And now there is additional clinical studies ongoing with NM, and there's NNM and many more with NR. But still, clinical evidence is still needing to be developed, especially in the context of healthy elderly. So this is still kind of lacking a bit overall in the field. And of course, as I mentioned, also the cost and the mechanism of uptake conversions, particularly for NNM, are still under debate. So more research is needed and ongoing for this. So since there is all this interest in developing or understanding NAD metabolism and which precursor is more important or which one could be applied for which specific condition, when I was part of the Nestlé Research Institute, we were actually focusing on cellular hallmarks of sarcopenia, as I introduced you in the initial slide. And what we did here is a study where we looked at a sarcopenic cohort in Singapore. So you can see here, lean mass, grip strength, and gait speed, which I mentioned before are the 3 clinical outcomes of sarcopenia. And they are significantly declined in the patients compared to the control. And what we looked at is muscle biopsies. When you look at muscle biopsies, we found that mitochondrial dysfunction was very prominent, actually, compared to healthy elderly. So again, healthy elderly were relatively healthy, but the sarcopenic patients were very impaired in their mitochondrial signature. And more importantly, what we saw is that NAD levels were significantly lower in the sarcopenic patients compared to healthy elderly. And most importantly, although it's a very small number of samples here, we still found a positive correlation between muscle NAD levels and muscle mass, grip strength, and gait speed, indicating that essentially that higher NAD levels in muscle would be linked to better muscle health overall. So this prompted us now to look at the serum from the same cohort. And so this is when I started my team in Nestlé Research and we led this project. We did metabolomics on the serum from the same cohort, and we looked in particular at vitamin B3 metabolites. So vitamin B3 metabolites are all these NAD precursors and intermediates that I showed you before, so important for NAD synthesis. And what emerged is that if we look at the metabolites that were changing, one in particular was significantly down in the sarcopenic cohorts, and that was this molecule called trigonelline, which at the moment, at that time, was not associated with NAD in humans or in animal species yet. And what's important about trigonelline is that as for NAD muscle, circulating levels of trigonelline were positively linked again to lean mass, grip strength, and gait speed. And even more interestingly, when we looked at the muscle transcriptomics, for pathways that were more positively associated with circulating levels of trigonelline, OXPHOS, so mitochondrial gene expression related to energy production, was the most positively associated pathway. So indicating that there's perhaps a functional relationship between trigonelline and muscle function and NAD levels. So this prompted us to ask whether trigonelline could act as an NAD precursor also in animal species and impact muscle health. So I'm not going to give you all the examples of all the work that we have done on this, but what we did, we test at least three models for this. We test primary human myotubes in vitro, and we test dose-dependently trigonelline for NAD levels, and we have also used sarcopenic versus control cells to see whether trigonelline would work equally in both healthy and sarcopenic cells, and it does seem to be the case. We have also used C. elegans as a short-lived organism for aging, and we could see that with aging, NAD levels decline, but they are significantly maintained or rescued by trigonelline treatment. And then when we went into the mice and we did a 2 hours gavage, and then for trigonelline, we looked at different tissues, we could see that NAD levels were significantly induced across different tissues, not just in muscle, for instance. We have done also mechanistic work. I'm not going to go in all the mechanistic work we have done, but just to give you an idea, when you work with cell lines, you can actually inhibit all the key pathways that I mentioned before. So you can use this inhibitor called 2-HNA to block the niacin pathway. You can block the salvage pathway for the use of NAM with the FK66. And then we tested NR as an independent precursor from these two pathways to assess the mechanism. And what you can see here is that if you treat cells with a FK66, so blocking the salvage pathway, you actually deplete NAD almost completely. But when you treat with trigonelline or with NR, you can actually maintain or even increase further NAD levels. But now you block also the press-handler pathway, NR still works perfectly fine, but trigonelline is completely blocked, indicating that trigonelline can actually go through the press-handler pathway as niacin. And in fact, trigonelline turns out to be just a methylated form of niacin, actually. And it's very stable in serum, differently from the other, let's say, NAD precursors, because it doesn't have the nucleotide unit. It's just basically deamidated form of NAM, so it's niacin with a methyl group. And more importantly, when we treated trigonelline, aged mice with trigonelline in the diet for 2-year-old mice for about 12 weeks, and then we looked at muscle fatigue or grip strength in these mice, we could see that aged mice, they lose muscle strength very quickly compared to young mice, but trigonelline was able to maintain or rescue to a good extent, and also the grip strength was maintained. So this supports the idea that trigonelline will sustain strength in the case of high-intensity contractions, and also grip strength overall. And again, I've seen this a lot. It's appearing on the market. I mean, of course, I'm not involved. But again, it's a new molecule, so clinical testing is needed. There's absolutely almost no clinical tests done for trigonelline as an NAD precursor, as muscle therapy for nothing. So this needs to be developed. Sourcing, understanding which source would be better, this is also critical. And this also prompted us to understand what other applications or properties trigonelline may have. And this leads me to the other part of the talk very briefly. So on the fact that we, as I mentioned, kidney disease is very prominent worldwide, and in particular in the context of diabetic kidney disease. So without going into all the epidemiology here, but in collaboration with the Amsterdam University and also with Zurich and with Nestlé Research, we actually got access to a human diabetic and diabetic kidney disease cohort in Amsterdam, and we actually decided to assess, as we did for muscle sarcopenia, whether trigonelline would change in this cohort. And what we observed is that if you compare early diabetic kidney disease in the serum or in the urine, trigonelline levels actually also significantly decline, similar to what we saw for sarcopenia. And actually, trigonelline levels correlate negatively with albuminuria in this case, both in the urine and serum, meaning that if you have higher trigonelline levels, you have less albuminuria, which means your kidneys are functioning better in this cohort. So with this in mind, this prompted us to look at diabetic mice which suffer from early signs of kidney disease. So they are essentially mimicking a little bit the human cohort that we saw there. And we gave trigonelline for about 8 weeks to these mice, and then we looked at things such as NAD levels, which are increased also in the kidney. So we have now muscle and kidney, but we have also data from liver showing that it can actually work in different tissues. And what we did here, we were interested in particular to look at the most metabolically active compartment of the kidney, so the proximal tubule, because that has a lot of mitochondria, so it's where the energy is required the most. So what we did spatial transcriptomics by using markers of proximal tubule and distal tubule and glomeruli, and then we performed transcriptomics on these particular areas, and we looked at what trigonelline versus control would be doing to these particular compartments. And if you look at the top pathways that are positively enriched in the tubular compartment, trigonelline actually was upregulating mitochondrial gene sets, again, a little bit to what we have seen in sarcopenia. And interestingly enough, it's also downregulating things related to inflammation, immune system, and so on. again indicating that it may have beneficial effects in promoting bioenergetics, while on the other hand also decreasing perhaps pathways related to immunity and inflammation, which is kind of new as well, and it requires more digging into it. And just to close with the last slide of data, to kind of really go precisely into the proximal tubule. So how does the proximal tubule benefit from trigonelline treatment? We have actually done a collaboration with the Zurich University, where our collaborators have developed a zebrafish larva model with a liver protein, which is fused to cherry. So cherry is a red fluorescent protein. What happens is that the liver will normally secrete this protein, and if you have a healthy kidney, it will be retained by the kidney and then back into the liver. But if you have kidney damage, the protein will be leaked into the water, and you can measure increased fluorescence. And that's what you see here. On the right, you can see that when you treat this fish with cisplatin, which precisely damages the proximal tubule, it's used to induce AKI, essentially. What you see here is that the leakage is increased, but when you treat with trigonelline, actually the proximal tubule in a way is protected, so there's less leakage and is significantly rescued. And again, this is mirrored actually by increased NAD levels with trigonelline, and even a decrease here with cisplatin, which is significantly improved by trigonelline again. So with this, I'm going to close due to time limitations. But essentially, the current status is that trigonelline levels decline in sarcopenic humans and in a DKD cohort, and that's associated with mitochondrial function. Trigonelline modulates NAD with a contribution to the Price-Sandler pathway, but we're looking at other pathways as well. And it may promote muscle resilience and kidney function preclinically, with potential applications for these tissues to be explored eventually in human cohorts. But of course, there are also remaining questions, which you can read here, and we can discuss further in the question session. Thank you.
Thanks for the speech. 谢谢 Professor Sorrentino 的精彩分享。请问现场的观众是否有问题要请教呢？
Hi, Vincenzo. Fantastic talk. I was wondering in the DKD model, you said it's through mitochondrial NAD TCA cycle. Did you look at all on whether PARP or sirtuin activities were affected by the increase in NAD?
Yeah, so normally we have not looked at it specifically yet. I mean, this is still early phase. I mean, we have done the spatial transcriptomics, and we have done a bit of validation then at the tissue level of the different, let's say, at least on the NAD mitochondria side of things. But normally, let's say during these kind of conditions, because there's more inflammation, fibrosis, these are NAD consuming pathways. So I would suspect that these sirtuins are also activated in this pathway. So I think for PARP inhibition, people have done on DBDB and high-fat diets showing, not in the kidney, but yeah, in the context of metabolic syndrome, that they are beneficial preclinically. So it's something that we could look even maybe as a combination, perhaps.